All right. Good morning, everyone, and welcome to our Up and Atom Undergraduate Research Symposium session. We have four wonderful presenters for you today. We'll talk about their research work that they have been doing throughout this year and possibly beyond. We'll have about 15 minutes for each session as each student will present. While they're speaking, because this is a webinar, you as attendees, the way that you can interact with the presenters is you can use the Q&A function that you should find at the bottom of your screen. You could submit a question and our presenters will either answer it during the presentation. We will also have a live Q&A after all four presenters have gone. So you could actually then also have the opportunity to have your question answered, ask follow-up questions, or have a little bit more of a discussion. And then it'll be an opportunity for you to perhaps unmute and we can have a little more of a chat. Otherwise, please sit back, enjoy our wonderful presenters. We're so proud of them and all of the hard work that they have put in. If you have any issues, you're also welcome to put those in the Q&A as well. And one of our moderators will try to address any issues you might need help with. So without further ado, may I call on one of you to go first? I don't know that we had an established order. Is anyone ready to rock and roll here? Haley, can I pick on you? I think I gave you a little bit of warning.
Yeah, I can absolutely go first.
Wonderful. All right, take us away.
All right. All right, cool. So, hello, my name is Haley Rice. I am a third-year undergraduate studying data science and biology here at the University of Oregon. For the past year or so, I've been working with the DeRose lab to essentially synthesize and test a lot of Oxaliplatin derivatives, specifically through modification of their 3,4 position on their carbon ring. So Oxaliplatin is one of the multiple chemotherapeutics that the lab works with. It's a platinum-based chemotherapeutic. And so a little bit of general history on platinum-based chemotherapeutics. The first one was actually discovered by Loretta Van Camp and Barnett Rosenberg, and this was during their research on E. coli. And so essentially, they had observed that the composition of a dividing cell looks very similar to the ion filings in a magnetic field. And so they had wanted to discern whether or not an electric field would also have an effect on cell division. So to do this, they added platinum electrodes to their solution with the E. coli because it was believed at the time that platinum didn't have any biological effects. Turns out this is not necessarily the case. So they actually unintentionally created cisplatin in their solution. And this was causing all of their results to end up with these extremely long strands of E. coli. We're talking like up to 300 times their typical length. And this was essentially because the cisplatin was blocking the cell's ability to divide. So they did further research on this topic and were trying to identify, what is causing this outcome. And once they were able to figure out that it was the cisplatin, they started researching its potential as a chemotherapeutic, which it is an effective anti-cancer drug. It ended up becoming one of the first FDA-approved chemotherapeutics in 1978. So following its discovery and its approval, there's a lot of additional research looking at other platinum compounds to see if any of them could potentially help fight against cancer. So this resulted in carboplatin and oxaliplatin both also being approved in 1989 and 1996 respectively. And you can see all of their compound structures in this top right corner here. So generally how cisplatin operates is that it's injected intravenously, so into the bloodstream, and the cisplatin has chlorides attached to it. Now the chloride concentration in the cell is lower than in the bloodstream, so these compounds are transported into the cell where essentially the chlorides break off and are replaced by water molecules. And so this allows the cisplatin to then bind to DNA in the nucleus, and this actually leads to DNA damage and eventually leads to cell death. So until 2017, this was how we believed that all platinum-based chemotherapeutics operated. Turns out this isn't actually the case. So a study came out in 2017 detailing how a subset of platinum-based chemotherapeutics are actually operating within the nucleolus and are killing cells via ribosome biogenesis stress. And so this group of platinum-based compounds includes oxaliplatin, which is one of the 3 approved chemotherapeutics. So a little bit of context for this: the nucleolus is essentially just a subcompartment of the nucleus, and one of its functions is that it's the site of ribosome biogenesis, and ribosomes synthesize proteins. So this paper was able to identify that certain compounds, such as oxaliplatin, were actually targeting ribosome biogenesis and therefore hindering the cell's ability to successfully synthesize proteins. And this actually can be observed some in that a lot of cancer patients are actually observed to have enlarged nucleoli. So this is a potential target that we can look at to hopefully develop, better refined cancer therapies. So sort of in summary, carboplatin and cisplatin cause cell death via DNA damage, while oxaliplatin causes cell death via nucleolar stress. So in light of this discovery, we're aiming to better identify what structural characteristics are causing oxaliplatin to trigger nucleolar stress, because ultimately we don't really know what's happening on a molecular level. We don't know, what oxaliplatin is interacting with, where exactly this interaction is happening, etc. So to study this, we do an assay with nucleophosmin. This is also known as NPM1, and that's essentially just a pathway marker to indicate when nucleolar stress is occurring versus when it's not. So as you can see in the no treatment and cisplatin images, NPM1 is typically concentrated in the nucleoli, so it's sort of clumped into specific parts of the cell. Comparatively, when there is nucleolar stress, so for this example in this oxaliplatin image, it actually translocates into the nucleoplasm, so it sort of spreads out into the surrounding cell when the cell is stressed. And so this is a really good visual indicator of when nucleolar stress is occurring versus when it's not. Then we can actually take cell images like these and then quantify this data. So essentially, we can calculate a coefficient of variation. And essentially what the coefficient of variation is telling us is just the variation in luminescence of the image. So a stressed cell is going to have a lower coefficient of variation because, and I mean, this intuitively sort of makes sense, right? Because a stressed cell is going to have NPM1 more uniformly spread out throughout the cell, so there's going to be less variation in luminescence. Comparatively, an unstressed cell is going to have a higher coefficient of variation, so it'll be closer to about 1 or so. So using these tools, we've been able to successfully synthesize and identify multiple compounds that cause nucleolar stress. We've also been able to identify that ring structure, hydrophobicity, and directionality are all confounding variables regarding the induction of nucleolar stress. One compound on this slide that I'd like to bring to your attention is docplatin, and I've included a larger picture of its structure down at the bottom here. This is a derivative of oxaliplatin, and essentially a lot of the research that I've most directly been involved with in the lab has been researching how we can manipulate this ring structure that docplatin has while still creating compounds that cause nucleolar stress. And so this is with the ultimate goal of creating an azide derivative that would create a clickable compound, and I'll talk a little bit more about the significance of that later. But during our process in attempting to synthesize this, we realized that one of the synthetic intermediates for the process, docacetamide, which you can see on the slide, actually does not cause nucleolar stress. So we're worried that something about its size in that location might be hindering the induction of nucleolar stress. And so we decided, let's take a step back. What's the simplest substitution we can do? And so we started working on syntheses for methyl substitutions in both the equatorial and axial conformations. And a methyl group is just a singular carbon surrounded by hydrogens, and you can see how the equatorial and axial derivatives differ ever so slightly in their conformation structurally. So this is what the syntheses routes look like for both the axial and equatorial derivatives. They're, overall pretty similar, which makes sense. They're pretty similar products that we're creating. They're just slightly different in their conformation. So this is essentially all of the syntheses that we worked on. And once the products were created, this is what the NMRs look like. So while there is a slight concern about minor products in our products, which would just be the other conformation, right? We're not worried that it's going to affect our data at all. As you can see, both of these NMRs are distinct. And so any minor product that is present is in small enough quantities that it's not detectable by proton NMRs. So it's not going to be in large enough quantities that it'll be able to cause nucleolar stress on its own. So we're not worried about it affecting our data at all. So once these compounds have been synthesized, we need to test how are they affecting the cells. So to do this, we introduce each of the compounds to A549 cells. These are non-small cell lung cancer cells, and we ran 3 trials with cells for each of the compounds. So as you can see in the images, both the equatorial and axial conformations and derivatives do in fact cause nucleolar stress. Docene over in the corner here, which essentially just is missing the hydrogen that would be in that location. There's a double bond instead, also causes nucleolar stress. But you can see how these images differ from our no treatment control, right? It's still the NPM1 in the no treatment is still very congregated in the nucleoli compared to how it's spread out in the other images. Furthermore, this is what the coefficient of variation data looks like for this research. Both the equatorial and axial derivatives, again, have lower coefficients of variation by a notable degree comparatively to our no treatment control. And each of these dots on the graph, just for reference, represents a singular cell in the trials. And this was a little bit surprising because we had sort of anticipated that the axial derivative wouldn't actually cause nucleolar stress, and this is because the docacetamide, which we had synthesized previously, was also in an axial conformation. But the fact that both the axial and equatorial derivatives do cause stress is really exciting because it essentially demonstrates that this ring structure has some threshold of tolerance, right? There's a little bit of wiggle room where we might be able to add on to it successfully. And so moving forwards, we sort of want to probe this threshold of tolerance further and determine, how far out from this ring structure can we build before it'll stop inducing nucleolar stress. And so we're starting to work on syntheses for both the ethyl substitutions in the axial and equatorial conformations. And again, this is with the ultimate goal to identify a potential location in which we can install an azide handle, which essentially would just allow us to make the compound clickable. And this would be really valuable because, well, first of all, we don't have any clickable compounds in the lab that currently also cause nucleolar stress. But clickable compounds would allow us to track the compound via fluorescent click addition. It would also allow us to do pull downs, which would essentially help us identify what biomolecules the compound is interacting with. So these tools would be really helpful because it would allow us to become better informed as to what oxaliplatin and other compound derivatives that function similarly and cause nucleolar stress, what are they interacting with, where is this interaction occurring? And it would allow us to become better informed to hopefully develop new protein compounds to better increase cancer treatment and its success. So finally, I'd like to just acknowledge all of the lovely individuals in the DURA's lab. Kristen McDevitt especially has been a fantastic mentor and very patient with all of my questions. And I'd like to also thank the Knight Campus Undergraduate Scholars Program for connecting me with this research in the first place and helping with funding. So thank you.
Haley, fantastic job. Thank you so much for your presentation and wonderful work. And I'd like to remind all of our attendees, you can ask Haley and anyone else questions that you have during their presentations using the Q&A function, or you can hold them until the end and we can have more of a verbal Q&A. So I'd like to turn us now to Parker Morris for your presentation.
Yeah, absolutely. My video is locked. So there's that. Oh, there we go. Hi, everyone.
Hi, Parker, you're muted.
There we go. Okay. My work's on the effect of sterics and electronics on nickel-catalyzed allylbenzene isomerization. We'll get into what that means in just a little bit. I did this work in the lab of Professor Amanda Cook, funded by the VPRI fellowship on campus. So there we go. So when we think about isomerization, it's important to consider the substrates. Allylbenzene is this substrate that I have shown here on the left. And let me get laser pointer here. So this is allylbenzene, and it's this really common starting material for a lot of different products. And it has this terminal alkene, which is pretty easy to install, but not very useful in terms of chemical synthesis. So what would be great is if we could isomerize this alkene, this terminal alkene, to an internal alkene, as I have showed here, which often adds value to the allylbenzene derivative. Three really important allylbenzene derivatives are isosafrole, isoeugenol, and anethol, as I have shown here. And these are chemicals that are made on the ton scale yearly, often up to the million-ton scale with regards to anethol. When you think about alkenes, installing that double bond is pretty easy. In sophomore organic chemistry, we learn about reactions such as the Wittig reaction, where we take this carbonyl and form an alkene. What's tricky about this reaction is geometry. So this R group can either point to the left or point to the right. We call that the E or the Z alkene. Isomerization is a tool that will help us mitigate the issues plaguing alkene installation. So there's two sort of flavors of isomerization. We can either take a terminal alkene and move it to an internal alkene, something we call positional isomerization. Alternatively, we could take an E alkene and isomerize it to a Z alkene. And that's something we would consider a geometric isomerization. In industry, there's ways that we do this already. In industry, we focus on base catalysis, so using some sort of basic system to promote isomerization. This is good because you can use it at a really large scale, but the loading for base is really large. So oftentimes we're seeing 2 to 10 equivalents of base to starting material ratio, which is pretty unfavorable. And if you're running on a really large scale, that's quite expensive. Additionally, because we're using a base, the conditions are particularly harsh with regards to substrate scope. A base can react with a lot of different substrates. So having high pH is pretty unfavorable, and it limits what we can actually isomerize. Additionally, these reactions are often seen to be run at very high temperatures, often 300 degrees or more, which energetically, that's really expensive. It costs a lot of money and a lot of energy to heat something up to 300 degrees or higher. And then additionally, the last thing that plagues these base catalyzed systems is the low selectivity. So E to Z selectivity is only 5 to 1, which if you're losing a fifth of your product on the million-ton scale, that's extremely expensive and detrimental. Academia has tried to mitigate these problems by using metal catalysis. So metal-mediated catalysts, I have 2 shown here. They often focus on precious metals. So ruthenium, this ruthenium catalyst was seen to successfully isomerize alkenes. However, it also promoted side reactivity, which again is unfavorable. Alternatively, we have this palladium catalyst, which was seen to really effectively isomerize alkenes in high yields, low catalyst loading, and high selectivity, and greater than 90 to 1 E to Z ratio, which is significantly better than the basic system. That said, both of these rely on precious metals, which are sort of just that. They're precious and really expensive. So what I was focusing on in the Cook lab was using nickel as a cheap, earth-abundant alternative to these precious metal catalysts. When we think about catalysts, the first thing that we think about is the catalyst design. That's what we call the ligand sphere. And so I have shown here this nickel in heterocyclic carbon, which is a really tunable and modifiable ligand that we can use for metal catalysis. When we think about nickel NHC, as they're called, there's sort of 2 ways we can modify. On the backbone, traditionally, this has been seen to change the electronic character of the ligand. Alternatively, if we modify the nitrogen substituents, that generally changes the sterics, or sort of the size of the molecule in space. That said, what's sort of novel about our system is we imagine that if we increase the size of that backbone, and we increase it, and we increase it, we're now not only changing the sterics, we're also changing the electronics on the backbone. So instead of sort of just the traditional picture, we're now modifying multiple different pieces by changing the backbone, which is an easily functionalized part of the NHC. With that in mind, I hypothesized 4 target complexes that range in terms of steric character. So this unsaturated NHC is sort of the smallest, and then moving up the line to the methylated NHC, that's sort of the largest complex in the system. We can quantify sterics using this parameter known as the percent buried volume. When we think about percent buried volume, if we take the complex and sort of encase it in a theoretical sphere, the percent volume of that sphere that's occupied by the ligand, that's the percent buried volume. And so as your ligand sphere, or as your ligand increases, the percent buried volume increases. And so the complexes that I targeted have actually been synthesized before and seem to be catalytically active in different reactions. However, based on sterics, it was seen that they had different reactivity. So the Louis group synthesized these and quantified their percent buried volume. What they saw was that there was about a 3% difference from the unsaturated NHC up to the methylated NHC. So going from about 36.9 to about 39.1, I think this is covered by the 39.6, my apologies. And so there's about a 3% difference between the percent buried volumes, which when we think about it, that's not a huge difference, but they saw differences in reactivity between the unsaturated NHC and the saturated NHC, which corresponds to only a 1% difference in percent buried volume. So a 3% difference should show significant changes in reactivity. With that in mind, this project had three aims. The first was to simply synthesize these complexes. While the Louis group synthesized them in 2018, we were hoping to improve the yields of the synthesis steps, and they also used costly equipment such as microwaves, which are not available to all synthetic labs. And so if we could eliminate the use of microwaves, we would make these complexes, which are catalytically important, to be more accessible to labs across the nation. Additionally, we wanted to test these catalysts once they're made in allylbenzene isomerization reactions, particularly focused on quantifying the role of sterics on product distribution and overall yield. So that's what percent of the alkene are we going to get out, and then what is the E to Z ratio if we just react over about 16 hours. Once we have this data, we're looking to determine the role of sterics on the initial rate of reaction. So does having a larger ligand sphere decrease or increase the speed at which isomerization takes place? We can hypothesize on what might happen based on this catalytic system that is the proposed mechanism for allylbenzene isomerization. Starting with alkene coordination, I'll just go through this real quick. We go through 2,1 insertion, beta hydride elimination, and finally get this alkene dissociation, forming the final alkene product, where we've isomerized this terminal alkene to an internal alkene, and either a Z or an E isomer. Several hypotheses can be made using this mechanism as sort of motivation. The first is that the beta elimination step, that's this step here, will determine the E to Z ratio. We can imagine that if the ligand sphere here was significantly larger, it would be difficult for this R group to sort of point to the left rather than point to the right. So that might be the step that promotes E to Z selectivity. Additionally, increasing the sterics will increase Z formation. So this is the Z alkene, and like I just said, if it's pointing to the left as the E isomer would, that might be difficult if the ligand sphere is significantly larger. Finally, or not finally, but thirdly, we think increasing the sterics might decrease the overall yield by reducing the size of the coordination site in this alkene coordination step. If it's harder for this alkene to get to the nickel center and form that nickel coordination, then it would be harder for the reaction to begin at all, and overall yield would decrease. Along those same lines and by the same logic, increasing sterics, we hypothesize, would decrease the initial rate of reaction. Again, if it's harder to coordinate to the nickel center, it's harder for the reaction to begin. And so not only are we decreasing the rate, we're also decreasing the overall yield. These were the hypotheses. So now we can begin by looking at the actual work that I did, with starting with aim one, which is simply to synthesize the complexes. And I'm just going to run through these particularly quickly. It's not super important that you understand exactly how we made these complexes, simply that we were able to improve the yields over literature. So this was the synthetic route to reach the unsaturated NHC, which we did in 94% yield, which is comparable to literature. Taking this imidazolium chloride that we formed in the unsaturated NHC, we can functionalize the backbone using carbon tetrachloride to get to the chlorinated NHC. And we notice that the yield is particularly low. However, that's simply because rigorous purification was undergone, and it was important that we had only this compound isolated and no impurities that could affect isomerization reactivity. Moving forward, this is where literature begins to get iffy. So we notice that now forming the imidazolium chloride, where earlier it was formed in 80% yield, now it's only formed in 55% yield. And that seems low, but based on literature, we only saw 22% conversion in literature. So increasing that to 55% yield is actually pretty significant. We deprotonate that to get the methylated NHC just as we did earlier. And then finally forming the unsaturated NHC. This step here, where we go from this diimine complex to the saturated imidazolium chloride, this was seen to only be done in literature using a microwave. However, we were able to do it using refluxing conditions, triethorthoformate and formic acid, and achieve 99% yield of the imidazolium chloride, which is fantastic. So with all that in mind, we successfully completed AIM-1. We synthesized all 4 ligands and all 4 target nickel complexes. We can move forward to AIM-2 now, testing the catalyst for allylbenzene isomerization, specifically looking in this first AIM at the role of sterics on product distribution and overall yield. We do this in this generalized reaction here. So we take allylbenzene, react with low catalyst loading, only 5 mol%, a hydride source such as silanes, which are tunable, and another parameter that we could change in this reaction. And then Durin as a product, is an internal standard for determining product yields. We then expect 3 different products: the hydrogenated impurity, the E-alkene, and the Z-alkene. And I'll remind you that we increase the percent buried volume as we go from the unsaturated NHC to the saturated NHC. And so this is what this data is the data that we collected, which is sort of difficult to look at, so we can visualize it a little bit better using this graph. And so what we see was that with the unsaturated NHC, this IPR complex, we got high yield, 91%. And with the methylated complex, we also got high yields. So this sort of disproves any sort of hypotheses we had about the role of sterics on allylbenzene isomerization. That is, we thought that increasing the sterics about the nickel complex would decrease overall yield. But in fact, this is not what we saw. We saw that the smallest and the largest complexes had the exact same yield and the same E to Z ratio. If we look at these other 2 middle complexes, we notice that the saturated IPR complex had the smallest yield and smallest E to Z ratio. This was actually attributed to insoluble impurities that were not visible by NMR. Moving forward, we were able, now we can look at the kinetic profile. However, we decided to throw out the saturated complex due to its sort of strange and unprecedented reactivity that was attributed to impurities rather than actual nickel complex. So now we react in the exact same system, but we decrease the temperature by a little bit and take aliquots every 5 minutes over the course of 4 hours. And I'll remind you that it was hypothesized that increasing the sterics would decrease that coordination site and decrease the overall rate of reaction. Running this reaction, we noticed that contrary to the sort of lack of trend we saw in product distribution, there's a very clear trend with regards to the rate of reaction for these complexes. The largest complex, contrary to our hypothesis, reacted the fastest. The chloride complex reacted the next, and then the unsaturated nickel NHC had the slowest initial rate of reactivity. And we can quantify this by linearizing the first several data points. Thus, the rate of reaction for the methylated complex is about 1.2 times 10 to the 4th moles per second. The chloride complex, 4.5 times 10 to the minus 5th molar per second. And the unsaturated complex was 5.5 times 10 to the minus 6 moles per second. Relating these numbers to each other, that's the methylated complex about 3 times faster than the chloride complex and 24 times faster than the unsaturated complex, which is completely contrary to what we anticipated. So our conclusions were that we established facile and effective syntheses for reaching these complexes that are particularly useful in a litany of different catalytic reactions. And then additionally, we established precedent for increasing the steric bulk, leading to faster isomerization rate of reaction. Moving forwards, the first thing we want to do is purify this complex, this saturated complex, and get a kinetic profile for it to see if it matches the data. Additionally, we can imagine increasing the size of that NHC even more and seeing if there's sort of an upper limit to the effect of sterics on alkene isomerization. We can also think about changing the electronics a little bit more quantifiably. So because we didn't see any clear trends in reactivity with regards to product distribution, that must mean there's some sort of electronic contribution as well. We could quantify this using this sort of line of electronically withdrawing and electron donating substituents. And then finally, if we can run this reaction with the silane, we can actually do what's considered remote functionalization and tandem catalysis, where you first isomerize and then add silane to the double bond. And with that, I'd like to thank everybody in the Cook Lab and the VPRI and UROP for hosting me. And I'll conclude my presentation. Thank you all.
Parker, thank you so much. Wonderful work there on your presentation. Let's go now to Stacey for your presentation.
Can everyone hear me? And do you guys see the screen?
You're great. You're great, Stacey.
Awesome, thank you. Good morning, everyone. My name is Stacey Andreeva, and today I'm going to tell you about structural changes in framework materials that are enabled with dynamic bonding. So when we think about structural dynamics, the first thing that comes to mind is phase transition. Oops, sorry. Okay. And on the left is a simple phase transition diagram of water. The material responds to its surroundings and equilibrates to become most thermodynamically stable at particular conditions, like a phase change from solid to liquid. And when material undergoes a phase transition, sometimes this process affects its optical, electronic, magnetic properties, and even causes crystallographic changes. And that's why studying equilibrium is important to understand phase transitions, as they directly correlate with equilibrium of material properties and dictate how it will behave in response to temperature, pressure, magnetization, etc. And another important thing about structural dynamics from engineering standpoint and application side of things is that understanding how certain structures behave under certain conditions helps with optimizing the design to achieve superior performance. And phase change materials are also often implemented in electronics, in switches, data storage devices, and optical displays. And as molecular materials chemists, we study organic and organic systems, and of particular interest are metal organic frameworks, also known as MOFs, as they're fairly tunable and find applications in fields ranging from electronics to biological systems. But the main reason we care about MOFs is because of their dynamic metal linker bonds, which simply means that they reversibly break and reform in response to stress. And it has been long established that in order to form an ordered crystalline structure, the bonds will have to break and reform to correct for erroneous coordination and eventually achieve the most stable configuration. However, people don't really think of these bonds as dynamic after the formation of the structure. On the contrary, they like to think of MOFs as rigid structures. And so previously, the dynamic nature of metal linker bond was revealed from post-synthetic modification studies with one of the most well-studied frameworks, MOF5, in which the authors performed cation exchange and discovered that the zinc center easily changes geometry when soaking in a solvent and subsequently changing the metal identity. An earlier study from 2005 looked at a family of structurally similar coordination polymers that have reversible metal linker bonds, and they identified binding constants for different analogs that turned out to be relatively low, indicating that reversible bonds are dynamic and have low binding constants. And another example is temperature-induced metal ligand bond weakening in self-healing polymers, which was explored using Raman vibrational spectroscopy where Raman modes of terpyridine shift to lower wave number upon heating the metallopolymer, which indicates decomplexation or dissociation of the metal ligand structure. So all of this inspired us to establish a model that would be representative of MOF formation mechanism and allow us to derive quantitative parameters of this dynamic interaction. And with this goal in mind, experimentally, we decided to study the two most common families of MOFs. One on the left is a carboxylate-based MOF, and on the right is an azolate-based MOF. And the main difference between the two is the metal ligand linkage. In carboxylate case, it's the metal center is bound to oxygen, which are the red atoms, and in the azolate, they're bound to nitrogen, which are the blue atoms. But today I will just focus on one system, which is called copper BTC, also known as HKST1. And so to probe the dynamics of the metal linker bond, we turn to vibrational spectroscopy, namely VT drifts, which stands for variable temperature diffuse reflectance infrared Fourier transform spectroscopy, which is basically just variable temperature infrared spectroscopy for solid state powdery materials. And another thing to note about the system is that metal oxygen bonds usually appear at fairly low energies, which fall outside of the range of conventional IR instrument. But we hypothesized that the dynamics of the bond can still be probed by looking at the adjacent bond, so carbon-oxygen bond. And this can be explained with simple molecular orbital picture of the CO bond, in which the highest energy electrons are in the orbitals that are either antibonding or non-bonding, which can also be thought of as a lone pair that interacts with metal ions. And accordingly, stronger metal-oxygen bond interactions would also enhance the CO bonding by distributing the antibonding density away from the CO bond and cause a blue shift, whereas weak metal-oxygen interactions should cause red shifts. And here is the IR spectrum of this material. And so upon collecting data, we do see the redshift in the peaks attributed to the CO bond. And what we're looking for is the change in energy at which the CO bond appears. And what we expect to see is the lowering in energy of the CO mode with increased temperature due to phonon losing its energy by coupling with another vibrational mode that is metal oxygen. And this explanation is sufficient in terms of energy, but unfortunately it tells us nothing about what happens structurally, which is why we came up with equilibrium model. So basically, equilibrium model assumes that there are two states between which material is shuffling, that is tight and loose configurations. In tight configuration is when material is at lower temperature, which means that metal linker bonds are shorter and interactions are stronger. And that would be our higher energy component, whereas loose configuration is the complete opposite. And by taking the spectra of all temperature values and performing global fit analysis of the band, we are able to get the areas of peaks for each component. And area corresponds to population of certain species, that is the concentration of these species at a given temperature. For example, on this plot at 50 degrees Celsius, the red shaded area corresponds to loose configuration population, and blue is tight configuration. And we can see that even close to room temperature, the material is still predominantly in loose configuration. And conventionally, stability constants are reported as ratio of log of bound over unbound. And in our case, that would be tight over loose. And finally, once we derived the stability constants at all temperature values, we were able to fit the data to modified Van Hoff equation with changing heat capacity to get thermodynamic parameters like enthalpy, entropy, and heat capacity of the transformation for each system. And comparing results, which are highlighted with the red box, to know formation constants for metal carboxylate complexes, not frameworks, we get a log K value of 0.6, which falls within the range of available metal carboxylate stability constant values for common MOF metal ions, within the range of 0.1 to 1.5, which is highlighted with the blue box. And what this tells us is that MOF metal linker bonds are surprisingly labile, because the lower the value of K, the more species at this temperature point are in loose configuration, which overall contradicts the common perception of MOFs as being rigid. And from Van Hoff analysis, we also get these values for enthalpy, entropy, and heat capacity, suggesting that this process involves a slight endothermic barrier, which is overcome by increases in entropy, which just simply means that the change in configuration can be overcome by putting more energy into the system, which will make the constituents move faster. And so in conclusion, we successfully demonstrated that the adjacent bond to metal linker is an extremely useful characterization handle, especially for low energy metal linker bonds. We also established a working model that represents the formation mechanism of the MOF as a function of temperature. We showed the stability constants at room temperature are comparably low, which is indicative of metal linker bond liability. We also derived thermodynamic parameters of the interaction between the loose-tight species at various temperature points. What I didn't have time to talk about today were the results for other carboxylate and azolate MOFs, but briefly, I have a summary table of the same parameters we determined for copper BTC, and we see that the bonds that are expected to be stronger, so those of metal nitrogen or titanium oxygen and zirconium oxygen, which I just highlighted in red, also show larger stability constants and smaller thermodynamic parameters, which is indicative of slightly lower degree of dynamics compared to copper BTC. But the main takeaway is that metal ligand bonds in MOFs are dynamic, which is what gives rise to many cool properties and can help explain phenomena they didn't have a clear insight of mechanism before. And moving forward, we want to study more materials with equilibrium processes and show how their properties are impacted by dynamic bonding. For example, this framework exhibits a spin transition, which can be monitored by magnetic susceptibility or UV vis, since the spin transition also gives rise to an electronic transition. And I would like to thank everyone in my lab, especially Dr. Carl Brozek for his exceptional mentorship and kindness that helped me move through the project, as well as Hao Kun, Dr. Hendon and his student Kwa. And I also want to acknowledge the funding sources at U from Presidential Undergraduate Research Scholars Program and funding from the mini grant, as well as external sources of funding, and you all for listening. And I'd be happy to answer any questions.
Wonderful, Stacey. Thank you so much for your presentation. I do see we have a few attendees who have joined since the beginning. So I just wanted to remind everyone that you are welcome to add in any questions you have using the QA function, and definitely hang on until we get to the larger Q&A session where all of our presenters can answer questions together. And then Lillian, will you wrap us up?
Yeah, just give me 1 second to get situated. All right, can everybody see what I'm sharing?
Looks good.
All right, so my name is Lillian Payne, and I am so happy to be here today to talk to you all about the work that I have been doing in the Hendon Material Simulation Lab, where I have been working on routes for inducing photoaccessible metal states in zirconium metal organic frameworks. And so Catalysis, which is the acceleration of a chemical reaction by a catalyst, is absolutely central to the industrial processes that shape our modern world. The global catalyst market was valued at over $35 billion in 2020 alone, and catalysis is involved in the manufacture of over 90% of industrial chemical products. So the availability, the efficiency, and the environmental impact of catalysts are thus vastly important to the makeup of our world in the day-to-day. And here is just a little quick plug for something that I'm passionate about. Catalysis was invented in the late 1700s by a really interesting woman named Elizabeth Fulham, who isn't often recognized despite the enormous impact of her discovery. And she also discovered photoreduction, which is a process that's critical to the work that I'm going to be describing here today. So Elizabeth Fulham is a woman we should be talking about in history. And so as we're all aware, our current industrial processes are having an incredibly detrimental effect on our environment. And because catalysis is so important in industry, the catalysts used have a major impact on the environment. And unfortunately, common heterogeneous catalysts involve rare precious metals, the mining of which can have incredibly harmful effects. You can see on the left here how the vast majority of catalysts are composed of palladium, platinum, and rhodium, which, as we can see on the right here, are non-earth abundant precious metals. The energy required to power chemical processes is also important, and our current dependence on non-renewable energy sources is detrimental to our very future on this earth. But photocatalysis, which is the acceleration of a photoreaction, a reaction involving light in the presence of a catalyst, utilizes the renewable energy of light. And ideally, we could be able to harness the vast and renewable energy of the sun, although that is not always possible, as we'll see later. And photocatalysis is involved in industrially relevant applications, including water splitting and CO2 conversion. The most common photocatalysts, or some of the most common photocatalysts, are transition metal oxides. And among these, titanium dioxide is considered the champion. In the plot, you can see on the right here, light excites an electron from the valence band, which is like the highest occupied molecular orbital in a molecule, to the conduction band, which is like the lowest unoccupied molecular orbital in a molecule. And then we have a sacrificial reductant that comes in and offers an electron to plug that hole. And the gap in energy you see between this valence band and the conduction band is called a band gap. And this is the energy gap that an electron must be able to cross to be promoted to the conduction band. And for a material to be considered a semiconductor, this band gap must be narrow enough to absorb light in the visible range. So for a photocatalyst to be considered a good photocatalyst, we generally have four properties. We have narrow band gaps, we have our valence and conduction band potentials aligned with reaction potentials, we have long exciton lifetimes, and hopefully we have the ability to modify our structures to be able to select for reactivity and selectivity. And titanium dioxide, our champion, has a wide band gap and cannot actually absorb light in the visible region. And its rigid lattice is also a barrier to structural modification that we could potentially do to modify this band gap. And here, once again, we're talking about metal organic frameworks, as Stacey introduced earlier. And metal organic frameworks are a class of highly porous materials composed of inorganic moieties, referred to as secondary building units, linked in space by organic linkages. Benzene dicarboxylate, which I have shown right above here, is a common MOF linker. The inorganic secondary building units often take the form of metal oxide clusters, which are similar to the transition metal oxides that I was discussing previously. MOF construction from separate inorganic and organic components make them highly modifiable, which, as we discussed before, is desirable in a photocatalyst. But an issue that arises with MOFs across the board is that they are commonly insulators with large band gaps. But titanium dioxide has a fairly large band gap too, right? You can see over here. So this issue isn't an unfamiliar one. And what's great about MOFs is that they are so modifiable, so structurally modifiable, that maybe we could do something about these large band gaps that they have. And MIL-125 is a titanium 4 MOF made of titanium oxide clusters and benzene dicarboxylate linkers. And it is in its secondary building unit structurally similar to titanium dioxide, and it is actually photoactive. So maybe this MOF has promise for photocatalytic applications. And what people have shown in experiment is that MIL-125 actually shows photoactivity in proton-coupled electron transfer reactions and linker to metal charge transfer reactions. And in both of these reactions, light excites an electron from the valence band, which is like our highest occupied molecular orbital in the molecule, to our conduction band, like our lowest unoccupied molecular orbital. And this electron comes from a hydrogen or from the linker in a ligand to linker to metal charge transfer reaction. And we want long exciton lifetimes, as stated before. And this arises from the exciton, the electron, and the hole it leaves behind being spatially energetically separated. And in MIL-125, this separation occurs. The plot that you see here to the right is a DOS plot, and the peaks all correspond to states in the material. You can think of them like molecular orbitals or corresponding to molecular orbitals, and we're indicating by color the atomic contribution of each peak. And so our conduction band up here in MIL-125 and in other titanium 4 MOFs is metal dominated. So these peaks are all by and large composed of titanium. And this is true even for MOFs that have been transmetallated to include titanium. And these photoaccessible D states allow for the desired exciton separation, as the titanium 3 titanium 4 redox couple leads to stabilization of the new state through an intervalence charge transfer. So now we have a MOF that is photoactive and has these desirable qualities for photoactivity. But how do we access these desired electronics in other MOFs? And unfortunately, the favorable metal domination of the conduction band that we see in titanium MOFs is not universal for other D0 metal organic frameworks. On the right, you can see the density of states for UIO66, which is a zirconium 4 MOF with a really similar structural topology to MIL-125. But unlike with MIL-25, we can see that the conduction band is dominated by these organic carbonate oxygen states instead of these zirconium states. And so when we model proton-coupled electron transfer on UIO66, we see a new state localizing on a linker orbital instead of on a metal orbital. And this isn't good because we're not going to get the stabilization that we want through intervalence charge transfer, and we're not going to get the long exciton lifetimes that we want. But you can notice that if you look just a little bit above the conduction band edge, you see some zirconium orbitals right up here. And so that makes us think that if we were able to destabilize our linker orbitals so that they went up in energy, then our zirconium states would now be our lowest energy unoccupied states, and they would be accessible. And so one of the simplest ways we thought to destabilize our linker orbitals would seem to be to remove their aromaticity. And so zirconium CDC is a MOF synthesized with the same structural topology as UIO66. You can see here that it's really, really similar to UIO66 on the left. and it has the same zirconium metal secondary building unit, but instead of a benzene dicarboxylate linker, it has a cyclohexane dicarboxylate linker, and so it is non-aromatic. So we have pretty much the same MOF, but the linker has been destabilized. And lo and behold, when we model our zirconium CDC, we see that the organic linker orbitals have been lifted, and our conduction band is now centered on these metal orbitals. So we have UIO66 in the middle of this DOS plot right here, and its conduction band is linker dominated. But when we move over to zirconium CDC, while we have a much larger band gap, we now have access to the zirconium 4 states. And when we go to model our proton-coupled electron transfer process, we see that in zirconium CDC, our added electron is indeed now localized on the accessible zirconium states. So we would be able to see our stabilization from an intervalence charge transfer. And this would give us the long exciton lifetimes that we desire. And maybe a more intuitive way to look at this for people who aren't used to looking at these DOS plots, we can see here in the modeling of the electron density of these particular states, of these newly added highest occupied states. So in UIO 66, our newly added electron is localizing directly on the linker, a little bit easier to picture than just the little organic pink or peak in our DOS plot. But in zirconium CDC, our added electron is delocalizing all throughout our inorganic secondary building unit, which is going to give us the stabilization that we desire. So you can see in the little bit that I've shown today that although only some MOFs natively present the desired electronic characteristics for photocatalysis, we can modify linker characteristics and end up giving the desired electronic properties even when the MOF doesn't natively show them. So we've been able to expose linker orbitals that we want to be photoaccessible, even though our MOF topology originally did not have these accessible. And this is just a little piece of the picture of how modifying one or both of the components of a MOF can modify the electronic structure. And in the future, we want a more complete picture of how the structural characteristics and the electronic properties in MOFs are related. And one of the first steps to doing this is to isolate the effects of linker structure in the MOF electronic structure. So what I've been working on in the future is in a series of really simple MOFs, being able to understand exactly how different simple modifications to a linker, like the modification we did today, is going to be affecting their electronic structure. And I'd love to give thanks to my entire group, particularly to my mentor, Chris Hendon, and my mentor, Jenna, who helped me throughout this project, and also to the now Dr. Austin Muraz, who helps me with a lot of the computational details, to Kwa and to the undergrads, and also to our funding, and particularly to our computational resources that allow me to do this work, the Tilapia, Stampede, and COAS supercomputers. And that's all we have for now, I guess.
Wonderful. Thank you, Lillian, for your presentation. And just a huge round of virtual applause for all of our presenters who shared with us today. Wonderful work. So now we have about a half an hour set aside for a Q&A session. So if you are an attendee and you would like to ask a question, you can either submit it through the Q&A function, or you are welcome to raise your hand. You should have that function as an attendee, and then we can unmute you and you can ask your question verbally. So we have that power as well. The other thing I would encourage is that all of you, of course, are colleagues, and perhaps maybe this is the first time you've met, or maybe worked together before. So I would encourage, if you have questions about one another's research, this is a wonderful place to also kind of cross-pollinate and talk about your projects with each other. Or if there's just something you didn't get to, or a question you wish people would ask you about your work, you're also welcome to mention those things as well. So attendees, send us your questions. Awesome, we got a nicely done everyone in the Q&A. And Dr. Leslie Coonrod is also our other moderator. So thanks for all of your work behind the scenes and for keeping us all on track tech-wise.
I have a question actually for all of our speakers today. First, I want to say, yes, excellent job. Thank you for sharing your work that you've been working so hard on. I know these projects are often, often don't get to talk about all the hard work that went into them. Sometimes there's a bit of a struggle. And with that in mind, I'd just love to know, taking a moment to reflect back on the past, however long you've been working on the project, what might you approach differently now that you're a little bit older and wiser?
I can go. One of the things that was most difficult for me in my project was actually synthesizing the target complexes. And we actually went through, so the ones I have shown are sort of like the second generation. We were trying to make actually a different complex. And so I guess, if I could go back in time, I'd love to start with the ones that we actually made. We wanted to make something that was completely new. And that's difficult because it's new. And so it would have been nice to start with the ones that I had shown and then maybe try and use reactivity that we learned there to make something new. But yeah, it worked out in the end, but there was some stressful weeks where things were not working and syntheses were not completing. So yeah, I'd say that was maybe something that I would change going back, but it's one of those hindsight 2020 things. Like, really couldn't have known that until actually doing it.
Yeah, I had similar issues where we had some complications with actually successfully doing our synthesis. So probably it would have been nice to figure out what exactly we were doing not quite right to be able to kind of sort of streamline that process from the get-go. It was mainly some timing issues. But yeah, it also would have been nice to maybe just this whole research process was very much a learning process for me. I enjoyed the research a lot, but chemistry isn't necessarily something I had like a really strong background in when I started the research. And so it would have been nice to have a little bit more of that contextual understanding when I was starting the research initially.
I think something I learned from this project in particular, but from all of my projects, and I mean, it can be a little bit harder to explain stuff just because theory is not something that people are as familiar with, and a lot of people aren't as familiar with computational chemistry. But just when you have a problem, before like trying a million things to fix it, just going into the literature and realizing that, oh, other people have probably had this problem and probably found out a smart way to fix it. So before I use all of these resources to like try to just change something and it'll work, just going and reading a bunch of papers was a really, really valuable lesson I learned from this experiment.
Oh yeah, absolutely. My advisor used to say, what was it? 2 weeks in the lab can save an hour in the library. So we need to learn to switch that, take our time and absolutely check out what's already been done, what's already known. Absolutely.
Stacey, I don't want to cut you off if you had an answer to that too, so feel free to hop in at any point or not. But I had another question. So I imagine on some level, at least, that all of your research projects were affected by COVID. Perhaps if you weren't able to work in a lab, or if you weren't able to have some of the on-campus or in-person experiences you might have been expecting. But I think also it's opened up so many great opportunities like this for people to join in and to hear about research. So I'd love to hear about if your perspective on sharing and science communication has changed with some of the changes in our world, or even if just the circumstances of this past year affected your research in ways that were interesting or unexpected or positive.
For positive impact, I would say that definitely having the access to like webinars that are going on like around the world that I've been able to go to just because of the virtual format was kind of a good thing.
Yeah, I was just going to echo that same sentiment. I feel like I'm a fourth year, so I just defended my thesis on this project, in fact. And yeah, thank you. I was able to invite people like extended family that were not, that aren't in Oregon, to come and watch. So that was really nice. But our lab, I didn't feel like was dramatically affected by COVID within the first several weeks. Like the first, I say several weeks, like 3 months really. No one was in lab, right? But we were able to do that book research, right? We were able to do the literature searches and find stuff that's been done and figure out sort of what we know and what we don't know. So that was sort of nice. But I think, overall, we're not looking at how to solve COVID really so much, right? So it wasn't dramatically affected, in the later months.
Yeah, I think sort of the online format of being able to communicate science and such has been really nice. It's something that's made it a lot more accessible, and something that, talking in this format is something I'm a lot more comfortable with. So it's been really nice to be able to engage with science and other people who are interested in STEM in that way. Definitely as far as hindrances, it was a little frustrating just because, especially with limitations on the number of people who could be in lab at certain times and scheduling that. And then also, I'm sure this is probably an issue that people in this group specifically ran into a lot with like syntheses, right? That's very much an in-person type of research. There's not really much you can do about that through a computer screen. So that was a little bit frustrating as far as being put back a little bit time-wise. But also it's not research that I'm personally involved with, but there are some people in our lab who are looking at some stuff that was COVID related. So that was kind of interesting to hear about as well during lab meetings and whatnot.
I'll pop in with another question. I mentioned this a little bit before, but I would love to hear it. What is something that I imagine you've done your presentations at least a couple times and talked about your work with other folks? What's a question that you always wish people would ask, or like something you wish you could dive into a little more, but no one ever asks you?
I think for me, again, just like looking at the syntheses, that part of the project was so significant. Like that, just making the complexes took the overwhelming majority of the time that I was working on this project. And then once I made them, it was pretty easy to run them in catalytic reactions. That took a couple of days. But just making the complexes was so difficult. And sort of, it's the least interesting part of the project. So it's hard to emphasize that because it's like, yeah, we made these things and also they're already now, so it's like sort of the least exciting part of it. But yeah, no one really asks about that. And there's not really a whole lot of questions to ask about that, but it's like one of the parts for me that's like, was the most impactful, finally making these complexes was like such a huge sigh of relief. But like, that's not really something that gets communicated in the project because it's sort of the least interesting aspect of it. Yeah.
Yeah, I would say that usually, like when you're giving presentations, you try to generalize on what your results tell you, and you don't really get to dive into like special cases that's like outside of the trend, which is usually, in my opinion, the most interesting to think about, like what's going on in that case. So, yeah.
Particularly in like presenting about theory in a field that a lot of times, I mean, it's built by experimentalists and people who do synthesis. I would like to field questions about the why I chose to do my experiments in the way that I did, because I'm doing experiments just in a different way. So yeah, just questions about the computational methods and why they were chosen as opposed to other methods, or why this particular way of presenting data was chosen. Those are always the niche things that would be super fun to talk about if they weren't super, super boring for everybody else.
Oh, Lillian, I would love to hear all about your computational methods being in the bioinformatics program. I love to hear about that. So you'll have to connect with me afterwards and I will listen to you for days on end.
I love hearing all those little tidbits. And if you think of anything else, definitely jump in. Leslie, did you have another question? I want to cut you off. I do if you don't, but
I do have a question. Yeah, it sounds like most of you are Parker mentioned just defending a thesis, and it sounds like many of you are sort of coming to the end of your undergraduate careers. Has this experience helped you figure out what you do, or maybe what you don't want to do in your next steps moving forward?
Yeah, certainly. I'll go. So, like I said, the synthesis was such a big part of it, and lucky for me, that was my favorite part of it. So, like running the catalytic reactions, especially the kinetics. I'm sitting at the bench top and I'm taking an aliquot every 5 minutes, and I'm sitting at the bench top for like 6 hours, just waiting, and then taking an aliquot, and then waiting, and then and so that part is sort of tedious, and I was like, wow, I don't love this, like this kind of stinks. But the actually making the complexes was so exciting. Like, sort of, it's like fancy cooking, I just love it so much. And so you add all these things together and they change color, and then you get a fancy crystal at the end, and everything is nice, or everything is not nice. But that's sort of the fun part, I feel like. And so yeah, moving forward, I'm going to graduate, right? And I'm going to go to grad school. And this project in particular made me realize that I wanted to do synthesis, and I wanted to do sort of like materials work. So I liked the catalytic aspect of it, but I liked making new things a little bit more than that. So yeah, this project was, really fundamental in changing and deciding what I'm going to do in chemistry moving forward.
I've definitely had, I think, a lot of difficulty sort of figuring out what exactly it is that I want to do. I've sort of bounced around a little bit. I've done some research with like the Neolab and done some neurobiology, stuff like that. So I've sort of dabbled some with research and classes and majors and whatnot. But I don't know if this has necessarily helped me like solidify exactly what it is I want to be doing, but I think that a lot of my experiences with research and similar things have at least like reassured me that I'm going to like enjoy some component of whatever it is I'm doing, right? Regardless of whether it's like my favorite subject or anything like that. Parts of the process are really enjoyable, and getting to work with other people is really exciting. So it's been nice in that way, that it's been a really reassuring, like hands-on experience for me.
Stacey or Lillian, anything either of you would like to add?
Not just specifically for the work that I presented today, but for in general, my time in the past almost 3 years now doing research at the U of O. I think I definitely have realized that doing research and doing the kind of research that I've been doing is what I want to be pursuing for the future, which has been really exciting and really a beneficial thing to be able to realize, and specifically realizing how exciting it is on so many levels, the process of understanding the tiny minute little processes that have effects on bigger processes that then have effects on the entire world through industrial factors and the way that nature works and stuff like that. It's a very vague way to say it, but just being able to figure out these tiny little minute things that have such a big sort of meaning for the world eventually is really exciting and is really cool.
Yeah, I was just going to say how, I don't know, I was thinking about it recently, like how I got into research in the first place. And it's like, do I like doing this because this is the first thing I started doing? Or do I actually like this because, I don't know, I like this? And I don't know, just, yeah, also after almost 3 years in research and after reading through more literature, you just realize that I do actually like this because you get to read about other fields and think, that's boring. I don't want to do this in the future. And
so Stacey, I think you talking about how you started out doing research is a really good link to the question I wanted to ask. So a lot of the work that I do is with pre-college students, so middle school and high school students. I love using these recordings with them because it's really fun for them to get a peek into what they could be doing. So I would love to hear sort of travel back in time and think about your high school self, or if you have a younger sibling or a cousin, what would you want them to know about research in undergraduate level, and what kind of advice would you give them about if they wanted to get involved, or even if they don't want to get involved, but you think they just might need a little more information?
So I was in a really fortunate position where I had a lot of sort of lab opportunities at my school. We had sort of a required science fair that everyone did freshman through junior year, and so you would learn about this scientific inquiry and the whole research process, and we had access to stuff to do research with. But I wish I had a better understanding of how collaborative actual research is, because I think doing a full research project on your own, unless you happen to go find someone to help mentor you with your research, right, is a little bit overwhelming. And still a really good experience. I enjoyed it quite a bit, but I really enjoy how collaborative actual lab spaces are, and sort of how intersectional research can be. And so I feel like I would have liked to know a little bit more about just the general experience of the other labs and how those spaces can interact with one another.
I think the most important, or one of the most important things that I would tell myself younger, and also just other young people who are interested in science and maybe interested in research, but I don't know, maybe are not confident in their abilities or in their place in the broader sort of science community, is to be humble and be grateful for being where you are and for accessing the opportunities, but also don't apologize for being there. Like, it's good to not think that you're entitled to resources or to opportunities, but also once you're there, you gotta own it and like be like, okay, I'm here now, I belong here, and I'm going to do what I want to do, just because nobody's ever going to take you seriously if you don't act like you are confident in the space that you are.
Yeah, also early on, I remember I was kind of shy to ask questions, so I would tend to like write them down, look them up on my own time, and I don't know, I feel like I could have learned a lot more if I just straight up asked what something means, and they would give me a more relevant answer than the answers that I might have found, or maybe that was a good thing. I don't know. Yeah.
Yeah, might echo what other people have said, but I think like the accessibility of research is pretty nice. Like, I think when I was in high school, I didn't recognize, I mean, I really had no idea what chemistry looked like. And so I think just, advising people to seek out those opportunities, and, if you want to do research, just do it. Like, like sort of what Lillian said, like, we want you, we want people to do research. And so, like, yeah, just be confident in the fact that, like, you can do research regardless of sort of like what your background is in STEM. Like, the lab wants to catch you up and wants you to be successful.
We have just a couple of minutes left together, so thank you everyone for your answers to our questions. Leslie, any other questions you have, or if anyone else has anything that they would like to add? We do have some time.
Not a question, but I just want to reiterate, it was truly a joy hearing you all talk about your work today. Clear you're all truly passionate, put a lot of hard work into what you've done, and I just want to say congratulations again, nice job. I'm sorry that we can't all clap for you, but I will clap for you. Those were amazing talks. Thank you so much for sharing with us today.
And I will say the same thing. I think be so proud of yourselves, go out, celebrate. I think maybe it's, I don't know, in the research that I've done, sometimes when you get to the point where you're presenting it and you're done and you're like, I am so tired of this. And it's like, so I don't want to project that, but sometimes, like, I just want to encourage everyone to be so proud of yourselves and recognize what incredible work you're putting out into the world, and just what an impact you're making on the world around you. So really impressive stuff. And I hope you all get the opportunity to celebrate and to see your colleagues and celebrate with them soon. So I think at this point we can wrap up. And again, thank you all of our attendees for coming and supporting, for our presenters. Again, this video in the coming weeks, the Undergraduate Research Symposium is going to be posting all of these videos to their YouTube channel. So you'll be able to pop back and check them out again. And until then, we have many more sessions later today that you can check out through the Undergraduate Research Symposium website. So hopefully we'll see you in another one. So have a great rest of your day, everyone. Take care.
Thank you, guys.
Thank you.
Thank you.